All right, it looks like we are at the top of the hour. So we're going to go ahead and get started with our webinar for today. Thank you again for everybody who's joining me. My name is Robin Komucheka, and I'm a technical application specialist with Denovo Software, and I will be hosting your webinar today. If you have any questions, please feel free to use that questions dialog. I want to make sure that what the information I'm providing today is helpful for you and gives you the tools that you need to start doing your analysis in FCS Express. So I really want you to get the most out of your use of FCS Express. And of course, if you have questions after today's webinar, you can always reach out to us at support@denovosoftware.com. We usually respond to questions within a couple of hours. We can answer those questions by email or provide trainings on screen sharing platforms. So please, please, please answer those or ask those questions so we can make sure that we give you the best support possible. So as you can see, FCS Express is not only a flow cytometry analysis software, but it can also provide the tools you need to analyze your image cytometry data. Today's webinar is a great start on exploring what FCS Express has to offer to you by introducing you to some of these tools and features and capabilities. Now, starting in FCS Express 4, we launched our image cytometry analysis solution and the presentation software package. Since then, the team at Denovo Software has continued to make improvements to FCS Express image cytometry to include more compatibility with various instrumentation and making continuous improvements to our analysis tools to make sure that we are keeping on top of having the most up-to-date tools for your image cytometry data analysis in our software. So let's go ahead and get started. So I'm going to start with workflow. So our software can handle data from instruments, the image cytometry, and we can also bring in images that are acquired on microscopes and other imaging platforms. So there is a lot of versatility here. So first thing you're going to do in your image data generation, of course, is acquire those images. Then we move to segmentation. Now, what is segmentation? What segmentation really does is it defines cell boundaries or populations of interest, and it allows us to then differentiate elements of an image and create distinct objects. Once we have these distinct objects, we can then segment the data and kind of extract additional information about light intensities and areas to really draw out the most information about these cells that we're looking at. So FCS Express Image Cytometry can read data files from a variety of free and paid segmentation tools. So if you were working from a microscope or other imaging platform, you would need to use this segmentation, some kind of segmentation software in the middle to create this segmented data files. But good news is we are able to import plenty of data files from multiple imaging platforms. So I'm going to kind of go over this image compatibility form now. Image compatibility table rather. And just a side note here, we are always updating and working with more manufacturers to increase this compatibility. So if you have an instrument or have some questions about if your data files will be compatible with FCS Express, please don't hesitate to reach out to us and ask those questions, or let us know that you'd be interested in having some compatibility with a given instrument. So those requests are taken very seriously, and then we reach out to those manufacturers and try to develop those relationships to get that compatibility going for you. So CountStar, or the ICE file format. There's phase holographic imaging, the Holo App Suite, Thermoscientific, the HCS Studio 2.0, Tissue Gnostics, and Yakagwa, and that's the ICE format. So we can bring those ICE files in directly. Nestsalom, we do bring in the NextStat files or the ICE files from that instrument, and then Amnis, the DAF or the FCS files can be brought in. Now, some of these things down below, Cell Profiler is one of the segmentation softwares. This is a free software and you can use it to segment your data. This will also segment out your images so you can bring in picture plots. We have the Chemometric, and that's an ACS file. We have Life Technologies, that's a Tally data folder, and that will bring in your data that way. We have that ISO file, IJOut. IJOut is also another segmentation software. And then from the Perkin Elmer, we have the Opera, Operetta, Acapella, Harmony, Columbus. These are Acapella folders or building blocks tab. Also from Perkin Elmer Vectra, we have the Inform software. So these are TIFF or text files. A little note there about the Vectra the Inform software, we are able to bring in the gate linking for Vectra Inform datasets, only currently available for exports from Inform software releases prior to 2.4.1. Raw data import and analysis is completely compatible with all Inform software releases, and then we are working on gaining that compatibility again to bring that gate linking in. So we have that in our plans. So once we have our segmentation completed, we've extracted those parameters or the feature extraction to grab those parameters and grab that additional information about a given file, we move into analysis. So analysis in FCS Express is really kind of a three-part task here. We build a layout by introducing those plots and gates. Then we display our original images to confirm, make sure that we're seeing the gates correctly and we're getting the populations of interest. We use those images kind of back and forth in with are those 2D plots. And then we do a report generation, whatever that reportable is, whether it's a PowerPoint, PDF, Excel. In our batch processing tools, we help you generate those reports very easily with a click of a button. So some of the topics that we're going to be covering today is the workspace interface, giving you a little familiarity about how things work, where things are in the software, inserting those picture plots and plots, understanding our data lists, and gating, looking at the gate view tool, data grids, how we insert those, what we can use those for, statistics and text tables, overlays, batch processing, and then saving a layout. Because the fantastic thing about FCS Express is once you've created an analysis strategy, you can save this as a template. So all the work that you've done building an analysis strategy, you save as a template, you get the data from your next experiment, you segment it, you bring it in, and you're able to reuse the work that you've already established here. So the main principles of flow cytometry and image cytometry overlap in many areas, such as taking those multi-channel fluorescence data and plotting that data in scatter plots and histograms, looking for those statistically significant differences and changes. And now that we can do this with image data, it kind of gives me a step up, or can give you a step up. So I was traditionally from a flow cytometry background, and getting used to image cytometry analysis softwares or using images, trying to integrate that into my analysis was a little bit difficult, but having one platform where I can do all the same things and use the same similar strategies for analysis has been really helpful. So with FCS Express Image, you can take your image cytometry data a step further by leveraging both those analytical tools in our flow cytometry software, and then using those multi-parametric data sets while relating the results back to those original images to confirm your results. So for that, we're going to get started here, and we're going to start looking at how do we perform this analysis. So I'm going to my sample analysis page here, and we're going to first kind of walk through these tabs just to give you a little introduction of where to find things and how to find different things in our software. So File tab, everything is very similar to PowerPoint, kind of gives you that similar PowerPoint feel. On our File tab, you'll find the save as, you'll find exporting options, and then most importantly, you'll find some user options. Now, user options help you define and customize this workspace, so when you come into FCS Express, you're really able to minimize the need to have to keep changing things and editing things. Once you find your style and the settings that fit you best, you come into FCS Express, it remembers those settings, and you just move on with your analysis. We have our home tab. This is kind of where we can do some building arranging. If we want plots to be the same size, we can align them horizontally, by widths, vertically. We can really set this up to have a nice presentation. In fact, as I'm doing today, I'm presenting from the software. On our Insert tab, you're able to insert a variety of things. So we have shapes and text boxes similar to what we've done in PowerPoint and pictures. And then we have our analysis options for our analysis that we're going to be doing with our plot types here. On our Design tab, if you're not sure how you want to set up your workspace, we do have some layouts to kind of help you get started and kind of give you that design inspiration and how you want to start aligning plots and kind of getting things set up. You can also change orientation. You can see here I tend to work right to left, so landscape really fits my style the best as I'm doing my analysis. You have the gating tab. Here's going to find all of your gating tools that you can bring into FCS Express to get that analysis completed. Then our batch tab. So this is really what I call the cherry on top of the sundae, if you will. Our processing tools are fantastic. What this does, after I've created my analysis strategy, I'm able to get those reportables right out of the software without having to take intermediate steps. I can create those PowerPoints, those PDFs, those spreadsheets, even internal spreadsheets if I want to do some charting and additional things downstream. So we've really worked hard to make sure that the software is working as hard as you are. And then when you need those reportables, it's easy to do with just a click of a button. And we will come back to this at the end of our analysis to show you how that works. On our format tab, we have 1,001 plus, right? Different formatting options. So if there's something that you are used to seeing in a given software, you just ask the question and we will help you get that formatting style in FCS Express. There are plenty of these. If I went through all of them, it would probably take all day, so I'm going to skip over that. Text tab, this is how we control our text boxes, text tables, and give ourselves some formatting ability of fonts and things. Our data tab is really where we can manage our data. One of the key things that I like here is the view header. This allows you to look inside any of your data files and find information. For our image files, that can be a little bit different if they are segmented, but it's still a useful tool. And then refresh, the ability to refresh your data at any time in case you change something, one of your settings. And then these are just some additional tools for your analysis. If you wanted to set up some concentrations or things, if you had some information to introduce there, you could do that. SOPs is more about standardizing a workflow for a user, or if you're sharing this template and you want everyone to be able to complete the analysis in the same way, you can tell them step by step how you want them to perform the analysis in FCS Express. And then on our view tab, we have some great things like a gate viewer, data navigator, layout viewer. And this is just to help you kind of organize and work through your data set and make sure that you know where gates are being placed, how your data files are being used, and what's happening on a given page, right? So if you have a multi-page workspace, this layout navigator is really going to help you kind of, okay, on this page, this plot is there, whatever. It helps you find whatever elements that you have added or objects or elements. So now getting started, I'm going to start on our insert tab and I'm going to insert a picture plot because, right, we're here for pictures and images and that's really my favorite thing to do anyway. I can grab that icon and then just drag it out in the workspace and then you'll see it will automatically open up a folder. So this is the folder that I happen to be working on before I got started. So it will automatically take me back to that folder. But I do want to take a moment and just show you all the different file types because if you were working with your data and we thought we had this file type selected, you move to your folder, you may not see your data. That's just because the file type is very specific for this given file type. If I opened it up to FCS files, you could see that I have TIFFs and other files in this folder. So if you ever get to a folder and you're like, oh my goodness, FCS Express has eaten my data files, don't worry, it's probably just the file type. Just make a quick switch, and you'll be able to see all of your data files there. So here, I'm just going to introduce one data file for this picture plot that I'm introducing. Click OK. And then one of my favorite things about FCS Express is it will bring in all of those other data files for me. I tend to be a little lazy sometimes and maybe not want to bring in all the data files. So this is a quick way to get everything from a folder in my workspace really quickly. Now we do kind of automatically put these in the order that they are in your data file or in your folder. But if you find that this order is not working, no worries. You can use your up and down arrows to reorder them. You can use your file name as well to kind of resort these in different options here. You can delete data files if there was a data file that you didn't want to bring in. All of those options are kind of there so you can help sort through your data and make the best of it. Of course, you can also add data using this blue plus icon, and it will take you back to the folder that you're using or let you navigate to a new folder. And then one of my other favorite tools here is this search icon. When I'm using this search icon, sometimes my folders, I can have folders inside of folders, right? So I have a big image data cytometry folder, and then I have multiple subfolders that I want to bring data out of, maybe because they were just different experiment days. This allows you to do that quite easily. So if I go to my desktop here and just navigate to my let's see, getting started image. If I were here in this getting started image folder and I clicked select folder and have this little include subfolders file checked here, it will go ahead and find all of the different data files. So this might take a second here as it parses through all of these different file types and kind of brings them in, but then you'll see like it's a quick way to load if I'm loading from different, many different folders beneath it here. And it might take a moment to think here. I have quite a few folders and information there, it looks like. All right, I'll go ahead and stop that. Oh, there we go, to stop so that this continues that search here. And then anything that was found before I stop that search is already going to be loaded. If I wanted to add these to my data list, I would just select them, use this down arrow to bring them into this bottom here, click OK, and I can load them into my data list. So after I have my first image here inserted, the beauty of FCS Express is that everything is fully integrated. So if I go to my gating tab here and insert a gate, I can start gating out a population. You just need 3 points here for our polygon gate. If you can't find that endpoint, no worries. If you click on the space bar, it will automatically close that gate for you. So I know that looks kind of wonky there. Looks like I got a little stuck between, but here I can just do, so I'll do a focal point. This is kind of the middle of my cells here. I can change this gate color, and then I can click OK and create this gate. Now, if I duplicate this plot here to insert another picture plot, I can also change my parameter. So this was segmented in 2 different ways for the images. We have the DNA and the cytoplasm. Now you'll see that gate is no longer visible, but that green population from the cells that are falling within this gate are still visible. As I move this gate around, it's also going to update those gating on the other image. So when I'm selecting cells out, I have that flexibility to work back and forth between 2 samples. And as I update anything, my plots are integrated, so it's going to update for everything else. Now, on this formatting, if I find, okay, it was great to see that gate, but I really don't need to see the gate for whatever reason, or I wanted to select gates. On our formatting window under gates to display, you can say no gates, so then it won't show any gates at all, or you can do only the gates checked below. So if I check my FOV gate, it's going to show. If I uncheck it, it won't. You have that ability to kind of customize what populations that you're showing here. Go ahead and close that. Now, if I pull this out, I can extract kind of a reverse, and now I'm just looking at that given population here on a plot. Additionally, other ways to introduce populations here is what I can do is grab a data file from the data list and drag it out into the white space here on my layout. Here I can select one plot type, I can select multiple, it's kind of whatever I need to start getting my analysis started. So I'm going to just do a colored dot plot here and start inserting that first plot. Now you'll notice there's a little bit of a size difference. When we go back to that home tab, if I need to make adjustments to my alignment, I can do that really quickly. I can align widths, I can align heights, and then I can even align it left if I want it to be nice and stacked here. Now changing these parameters is really simple. So just like we did for the cytoplasm and DNA, all we're going to do is click in the middle here, and then we can start seeing all of our parameters. So you're going to see all of the different parameters that we extracted during that segmentation. And you can see there are quite a few here. So if I was digging through, it might take me a moment. So fortunately in FCS Express, you can just start typing away, and you will reduce the number of parameters that are displayed. So we still have an area shape for quite a few, but we've also eliminated some of the other ones like the intensity. So we'll change that to area shape there. And then I will do mean intensity for our cytoplasm and click here. Now it can look like these dots are these events that we're showing are pretty small. If you see that, no worries. We can go to our overlays and increase the dot size here. So we can increase these events so that we can really see them. And now you see while we increase that event size, we're also already interacting with our other plot. We already see that this gate here. So if I wanted to apply that gate, I could drag this gate onto this plot, or I could go to my gating tab and just select it if I only wanted to look at populations that were falling in that gate. So we have that flexibility, that interactiveness to make sure that while you're doing your analysis, you're getting exactly what you'd like to get from FCS Express here. So here, I'm going to actually switch these. I actually put these, I'm going to, oh, I guess this is fine too. Going to create a gate here. We'll just use a rectangular gate. So if I select it, I just drag it in. And then here for this gate, I'm going to create a, I'm going to actually change this. I'm going to say small, dim cyto gate. So right now it's kind of on the larger side. So I'm going to just move it around and just change where this gate is falling. So we want the smaller and the dimmer, so maybe somewhere in here, right? So that's the nice thing also as you're working through FCS Express, you can update your gates. Oops, apologies there. You can update your gates and FCS Express will respond and update those plots as well. Take a moment here to get my FCS Express back up. All right, here we go. Looks like I already have a gate already inserted here. And I will do my picture plot as well. All right, so here I already have some gates established. So we'll kind of move forward here and just kind of pick up where I was leaving off just a moment there and increase this dot size so we can see these different gates. When I insert a gate view, you'll be able to see all of the different gates that I've created. And of course, as I update any gate, it will move and update in the workspace as well. So I'm going to go ahead and just, let's see what gates are here. If you're not sure where a gate has come from, so this one, the center FOV, that was definitely something I had drawn on image. In this workspace, if I'm not sure where a gate has come from, I can double click and open up some gate properties, and it will tell me exactly where a gate has been defined. So area, shape, area, and then intensity, integrated intensity for the DNA. Then when I right click, I can show that gate. And of course, I'll have that same flexibility that if I'm moving this gate, you can see in my picture plot above that this is going to update for me as well. So now gating, of course, we have many different gates that you can use and you can create these plots here. I'm going to just duplicate a few and update this for what we've already got established. So we can see our gate placements. Oh, if you do have to do a show gates, a good thing to note is I can recreate those gate text boxes. So by right-clicking, I can select recreate gate text boxes and then select all, and then I'll have that statistic here. So I'll go ahead and recreate this other gate text box as well. Then I will do a show gates here for my two populations, and then recreate all of those text boxes as well. So once I have a gating strategy kind of began, I can also integrate some other tools that can be very useful in FCS Express. And one of those is a data grid. So with this data grid, I'm able to insert a cell by cell information about what's happening for a given population. So what this does is it will give me all the parameters that I've collected. So right now it can look pretty overwhelming because we've got everything in there. No worries. This again is something that we can format backwards and kind of tell the software only certain elements that we want to see. So maybe we want to do area and shape, and let's see what else we used. The intensities for the cytoplasm and DNA, integrated intensity cytoplasm, and the DNA is right next to it. And then we can also show those pictures. So this is how, when we went back to that segmentation piece, this is what we had asked the software to do. We want you to define different cells based on different criteria. And then these are kind of the images that it extracted for each of these cells. So here you'll be able to see what those cells look like and how these cells are being defined and what's being used. Here at cell number 8, that's a really good visual here. We've got the whole cell here. Then you can see that DNA here in this next image. Additionally, if we wanted to add some gates to display, we can show what populations are falling in what gate. So it kind of gives us some additional information. Now this range gate that I've created is actually a gate that we can create from our data list here. So we can create a gate by right-clicking, clicking on create a gate, and then we can select a range. So we can remove, like if we were editing a gate, we could edit that range gate, select different gates, but it gives you kind of a flexibility. If I'm looking at these images and kind of going cell by cell and I want to determine another gate, maybe it's a gate that falls in my large bright nuclei and my and another given gate. Maybe it was the center FOV. I can create that range, and then I will be able to use that gate kind of moving forward, similarly to what we were building before. All right, now I forgot to turn off my auto save. So that's another feature to let you know that we do have. If you're working in FCS Express and kind of working through your analysis, we do have the ability to do some auto saves in case you need to step away from your computer or something comes up. You can have the ability to save your data and then keep moving forward as you work through your workspace. Let me see if I can escape here. All right, there we go. And now we just have this work. We have our data grid here. Now moving forward, I do want to kind of show you a few different other analysis templates that we have. And then we'll get into looking at different elements of how to do our batch processing, maybe do some overlays and kind of look at how other tools that we have in FCS Express can really useful for your data analysis. So if you were working from some plate data, you do have the ability to do some colony counting. So if I was creating something here and I'm looking at my information, so this has been segmented, it has been used by Cell Profiler. I took these images, I started defining different cell populations, and then I can do the same thing I was doing in the previous template, right? I've created these gates already. As I move these gates, you'll see that everything is updating, even that data grid. You'll see the images of my cells so I can look at what's happening for these actual colonies, and everything is still going to update as you're working through this. Now, if there was something that I wanted to do comparatively, you can use the data and do overlays. So I'm going to use the same data file because I think I'm only using one here. I can drag this out into my space here, click on the histogram. Oops, I actually had my picture plot selected too. And then here, just like if we were working in any other 2D plot, we would have all the different populations that we were looking for. So if we have masked red, we can introduce that here. We can also compare populations. So if I'm looking at a given parameter here, I can go to my formatting tab. Right now, I'm not looking at any gate, but if I wanted to look at red colonies, I could do red colonies and kind of look at what that is displaying on this given parameter. Now this might not be the best parameter, so maybe if I did subtracted red or find the parameter that would be best for my analysis, I could select that as well. If I drag that data file again, I can also add this as a new overlay. And then for that second overlay, I can tell the software, maybe I want to look at white colonies, right? Or whichever other large colonies, so I can see the difference here. So this color is in red. My black is that first overlay. So this is my red colonies alone. The second overlay is looking at those large colonies. Now with this, you do have some flexibility. If you don't like them kind of overlaid like this, we can move to our specific options and create them as stacked so that you can see them kind of separated and see what kind of, so it looks like maybe some of my large colonies are also following in those red colonies. We have like kind of a shared expression there. But that's overlays for you, and one of the ways that you can use them in FCS Express to kind of get the most out of whatever information that you're drawing out from these plots, or from your data analysis rather. Also, I'm going to go ahead and open a few different files here so we can look at batch analysis. And then I'm also going to open up for high throughput. So if you're using like plate data, we can use that and still insert images, get information here about your cells. One thing that's really nice about these heat maps is that they are, again, formattable. So if we go to our format layout here, we can do things like color levels. If we don't like a given color level, we can change that. You have a lot of flexibility in what colors you want to use, what's best for your presentation, what separates populations better. You can also change style. So you can use fixed range, you can use percentiles, you can use thresholds like mean standard deviations here, and then you can use thresholds. And these would be based on if you just wanted to do an on-off feature, you could set a minimum. So if it's above this threshold, it would be this color. If it's below this threshold, it would be another color, kind of giving you that on-off feature. Go to yellow-green here, and then we'll just go back. Also, if we look at this additional heat map here, what we can look at is we can do some really great things by giving radius parameters or radius statistic parameters, right? So what this does is designates the size of these wells so we can look quickly and be like, oh, okay, so not only is it on, but there's more happening here, right? Because the arithmetic mean of this population, it's fluorescing brighter, it gives you that scale kind of in another visual manner. Larger is more on, and then smaller is off, more off. All right. And this is kind of choosing parameters, looking at the different parameters that we've segmented out, and then moving these gates again. Again, as we're moving gates, things are going to be updating for us in the background. So if we look at this picture plot with the gates, as I'm moving things away or forward, I've only designated for a few, these two populations to be displayed, but you can see like not only is that picture plot updating, but my plot to the right there, and even my data grid. So if I'm showing something in a data grid, I can actually assign gates here as well. So here you'll see that I have my translocating cells already defined on this data grid. So I'm just looking at the shapes in the cells from this given population. You can also introduce any other cell type if you wanted to, look at whatever gates that you've created. We've tried to make sure that everything is as integrated as possible. So then while you're working through your analysis, you're not only able to update gates to make sure that you're gating the populations of interest, but then you can always reflect back and look at those images and make sure that they are accurate for what information that you've kind of derived from your analysis. So here on this last page, this is another feature. So with our spreadsheets, we can introduce great things like these curves. We can show some additional information in different ways. We can use charting tools, and we can use additional information to really bring home whatever the end goal is from our analysis. So on a lot of posters, we want to see what's different between our control and our test. These are elements that are already in FCS Express. So when we pull information into spreadsheets, we can also, create these dose response curves and present more information. You can also bring in figures, and we've recently introduced the ability to do links. So if there was something that you wanted to link to, maybe it was a given experiment protocol or some other publication, you can actually add those links into FCS Express as well. So you have a lot of flexibility in how you want to present or represent your data. Now here we're going to look at the report page. So this is kind of their summary page for their analysis. One thing that's nice is in these reportables that we're going to create next, you don't necessarily have to report out everything. You have the flexibility to report on one page, report all pages, to do things like compare things side by side and do the get the most out of these reports. So if I go to my View tab, because I have a few windows open, one of the little tricks is switch windows. It will allow me to move through my different things that I've already had opened. So I have this batch analysis layout file already open. I'll click here and it will move to that layout. So it's kind of a quick way to work through your data files. So here we're also looking for at some image data files. So this is from our AMNIS, and we're able to see those images here in our data grid. You can see the different images for the bright field and then for this parameter labeled DQ5 for channel 11. So we'll be able to see some comparisons of fluorescence as we move through our analysis. So one of the things that's really unique about FCS Express is that my data analysis is really the backbone for everything. What happens is that I am able to set it up for a given sample. So maybe I started with this sample number 1, and then I can move my data files through. And as I move through, you'll see that it's going to update with the information from a given data set here. If I move back, we'll be able to move back here. And let's see if I accidentally unchecked some of my parameters here. Not sure which ones I was showing here, but I accidentally may have disconnected these when I reintroduced my data from the CIF file. So what FCS Express does is when we introduce our DAF files, as long as they're in the same folder with our CIF file from the MNIS, it will bring in those images and you'll be able to report them in the data grid. Now, if they get disconnected, so if I had accidentally put my DAF folders in one folder and put my CIF folders in another, it won't be able to reconnect them. Also, nomenclature of the data file is really important, so you need to make sure that that nomenclature matches for each of the data files to make sure that when you're pulling in a data file, you're able to also pull in that CIF data file. So here, as you can see, when I walk through my data files here, I didn't update, this data file on my histogram didn't update. And we actually set that with great intention. So if I wanted to use a control here, I can set a file as a control, and in my overlays options, just tell the software not to change this data file during next, previous, and batch. So this can be great when I have, again, like this is my control file, and I want to comparatively look at this for different data files. So I would just drag it onto that plot again, add this plot as a new overlay. I'm going to see that it's going to match because that's the original data file that was on there. And then as I move to my next data file, I'll start seeing those differences between different data files and how that is expressing itself. So that's kind of one of the ways that we can use that. With our panels, we have updated our panels. So we have some awesome new ways to control your samples as well. So if maybe a couple of these samples were collected on a AMNIS to maybe gather different parameters, I can say, tell the software, I want to use 3 of these samples in different places through the layout and kind of create the ability to look at 3 different samples and maybe in 3 different spaces. Maybe I was looking at a certain cell type in tube 1, a second one in 2 and 3. with the same floors, or whatever that experimental design is, I have some flexibility. But beyond that, we have the ability to add stationary controls. This is great if I did have a set of control samples that I wanted to keep stationary on a given histogram and then be able to cycle through all of my different data files. So in this instance, if I had a panel of 3 tubes, right, we'll do 3 tubes here, and I had 3 controls. And we'll just kind of change these data files. Oops, let me give this a name here. Then I'll just kind of change these how I want to use them. And it's not necessarily true for the experiment, but this is a good demo here. So here at my top, I'm using these controls. So now anywhere that I use these controls, FCS Express will automatically know that I'm not going to be batching through these. It'll allow me to use these control samples as needed, and then it will be able to use these other data files individually, so I can pull them out and maybe do on a page histogram. So I'm looking at one data file to the next, kind of overlaid over and over for my different populations, so I can compare those controls to tests in different ways. That would be one way that you could set this up. Another beautiful thing about our panels is after you create them, if you don't need them, you can just uncheck them, click OK, and then it will go back and restore that individual data file approach that we're looking at. So just a quick visit, because we're going into batch processing in this template, is these iteration numbers can be very important. So you saw this one had already been set to a control for this initial histogram where I added the overlays, but 2 places where these iterations get grayed out or deactivated is usually in our spreadsheets or using histograms and overlays. And what that means is the software is trying to remember a pattern, and once it gets to a place where it can't repeat that pattern, so if I used data file 1 and 6, when I got to 2, because there's no 7, all these data files would be grayed out, just because it wouldn't know there wouldn't be a data file there to keep that same pattern. And we did that by design because our batch processing is the tool that walks our data files through to create those reportables. So for this, I'm going to take this plot and I'm just going to copy it and move it to page 2. Why I'm doing this is one of the things that I've really liked is the ability to look at a given population side by side for each iteration to kind of see what's changing, what has changed between different data files, and give you that beautiful presentation to look at samples. So now as I'm building up this batch presentation or my batch actions here, I can select the actions or the things that I want to create from a layout. So here I'm going to do a save per PowerPoint. Like we talked about before, you can have a very big layout with maybe 7, 10 pages and only report a few. So we can change that. We can use all pages, current pages, or select a page. For this PowerPoint presentation, I'll do one. I want the software to open after saving. And then here down below, we have some options. So we can tell the software that each iteration or each data file as it's listed right now is going to have its own presentation, or we can keep them all in one. So this first option is to save them each separately. This other one combines them. So you can just combine them. Additionally, if you're doing a longitudinal study where I'm looking at how things are changing for my cells over a given period of time, we can append these to existing presentations so that you don't have 42 different presentations that you're trying to bring all back together at the end of your experiment. So we're going to start with a new one for today, but you do have that option to append if that's how you would need to use it in the future. I'm going to open my folder here, and I will give it a presentation title here, and just click save. You are able to move to any folder that you need to create this element for, so you do have the ability to navigate there. I just was already in the right folder, so I can just click OK once I've created that. Now I'm going to save the second thing as a PDF here. So here I'm going to do page 2. And what I'm going to do is I'm going to tell the software a custom grid, and I'm going to crop it to the content. So crop it to that plot. And then I'm going to say, let's see, we have 6 samples. So I will probably do, let's do 2 columns by 3 here. Oops, by 3. And then click OK. You do have some ability to change which order that they show. So if you prefer vertical, so it'd be 1, 2 vertically, or if you would like it or 1, 2, 3 vertically, and then move to the next column to do 4, 5, and 6. Or you can keep it horizontal. It's 1, 2, 3, 4, 5, 6 as you move down the page. And then you can change the gap mode is also here. So click OK. I like to use the combine the output from all iterations into a single file. If it runs out of space, it will still move on to the next page, but it does it really nicely and cleanly. So I really like to use that feature when I'm using this option. And then I will put side by side here for my PDF. Click Save, and then Open File after saving. Let me make sure I click the right page there. Good, perfect. Now with our Excel sheets, we've now introduced the ability to do an internal spreadsheet. This can be fantastic if we're doing comparatives work. We need to report out our information for all of these samples. Maybe if we're even introducing some of those charts, the bar charts or pie charts. Remember, I had told you before that one of the things that deactivates your iterations here is a spreadsheet. So using this option to use an internal spreadsheet and having the software create it for us, it will eliminate that as an issue. We'll be able to create a spreadsheet here and then create those charting tools as well. Or we can do it export to just an Excel file outside of FCS Express. Same here, you can still append to an existing file if you're doing a longitudinal study, or we can create new ones as well. Click Save. Now, once I've dedicated where I want this to be saved, I do always open everything after saving, but we do have some options. So if I wanted to name my worksheets in a specific format, we can adjust here or use tokens. Tokens are just a cool way to say piece of information. We can use statistical information or the file names to report out spreadsheets names here or an experimental name. You can also tell the software where to start. So if you didn't want it to start in the first available row or column, you can actually designate, start row 1 and column A, or whichever works for your experiment. And then you can also tell it to write column labels or rows, or the column labels there if you need them. So after I've created this Excel sheet, though, initially it is blank. So if I do want to pull out information, I will need to drag and drop from the plots to start building this up. So again, we see that word token, and that's just a piece of information here. I can use the statistic token and I can pull out that file name. Click OK, and that will update our file names. Now, recently we have introduced the ability to customize statistic names. So if you would like to really drill down to a specific naming option, we do now have the ability to give you that flexibility to really define how you want things to name. So this is one place that you can do it kind of as a global expression, and your statistics will always, or your statistic naming will always follow this pattern. If you want something unique, you can also double click on any of the elements that you add, click on field, and then just text in whatever you would like this column name to be named, and clicked OK. So we have a couple of different ways that you can make sure that your columns are named in a way that works for you and makes sense. And then you can also just drag more statistics. So what I'm doing here is I'm building this for one data file, and then after I build it, FCS Express will do the rest. It will walk through all of my data files, and it will give me the information for each of the data files that it walks through this analysis. So I'm going to do number of events here and percent gated cells from plots. But before I click OK, I do also want to take a moment to highlight one of my new favorite additions, and it's this percent of all. Now, I know initially it looks kind of like, well, I mean, isn't that the same percent of all cells? It's actually not. With this gate, we can do a custom denominator. So any of the gates that I've created up to this point, even if it was a hierarchy that had, multiple generations, I could go back as far as I needed to by just changing this. And as I change that custom denominator gate, it will also update in the statistic window to tell me what I'm looking at or how I'm looking at this one population and what its relationship is to another gated population. So I'll go ahead and do number of events and gated cells here. And here I'll just adjust my statistic name. I'm pretty specific, so I like to do these manually. But again, you may just want to use those statistic options for naming your columns here. And so after I've built this out, you can either keep all of these elements or you can uncheck them. So if I came in here and today I didn't need that PowerPoint, I can uncheck it. I can also do the same with the Excel and just uncheck the whole Excel. Or if there was a day that I just didn't have a statistic available for whatever reason, I can uncheck an individual statistic as well. After I save this, this is all part of my analysis template. So I don't have to come back here and recreate any of these elements. It will be saved and I'll be able to just bring back those data files and or the new data set, put them in as long as I've segmented them all the same or they come from the instrument with the same parameters. I can drop them into this experiment and just pick up where I've left off, maybe move some gates around, do the kind of minor things to adjust my analysis appropriately, and then create these reportables. Um, so additionally here on our batch processing tabs, we have options. These options are super great in that sometimes, we don't need all of our iterations. Maybe we only need 3. We can tell the software we're only doing 3 and, adjust that as needed to give us the results that we need. We can also use pausing. So pausing is great if I'm working with a smaller data set. Maybe I unconditionally pause on each data file. If I just want the software to alert me if something is going on, maybe my viability is less than 60%, I can have the software pause if that condition is met. I can also flag iterations. So if there's a specific data file that's been giving me a headache all day, I can tell the software, hey, I need you to take a timeout on this data file so that I can see what's going on before we move on to the next one. And then you can also just designate. So if I know, looking through my data list, I have 105, maybe 105, and I just know that it's been a problem. I can just tell the software to pause on 105 or 105 through 107 if it was like a given sample set. And then, don't have to worry about flagging here. So I'll show you what that unconditional pause looks like. We'll click OK here and we'll run this batch process. So as it executes these elements, it will give you this run window. It will tell you like how it's creating what's been created. This is what the pause looks like. So in this pause, I have the flexibility to move any gates that I need to, move to different pages if needed, make any of the adjustments that could be necessitated as I'm walking through my data set. Now, like I said, this could be really convenient for 6 samples, but maybe more be a kind of a pain in the neck, but maybe only 2. Data 2, file 2 was the one I was kind of worried about. I could make some adjustments here and then just also tell the software to run to end is what I'll be demonstrating today. So I'll do run to end. It will create these elements for me and then they will open up for me. We'll see which files that they go to here. So here is my Excel sheet. I have each of my individual data files labeled there and the number of events and the percent of gated cells. Now this is an Excel file, so if there was something additional I wanted to add in that I hadn't done in the software, I can just pick up and keep working from here. I'll go ahead and close this window. Sure, we'll save that. And then this is my side by side. So I had told it 2 and then 3, so I could look at these 6 different iterations here, my 6 data files, and see what was changing between each data file. Again, super useful for a reportable if somebody just wants kind of the quick, okay, what has changed? What looks different between my different samples and my different populations? And then here, my PowerPoint is over here. I just had reported out that page 1. So we'll be able to kind of the same that we were looking at those iterations, the side-by-side iterations, I'm able to see the 6 different data files on different pages here to get that reportable. Now, I know I have talked a lot and I saw that Greg has been working overtime on questions over there. Let me see here. All right, so now what I want to do is kind of give you some of our tools. So I know I talked about our image format capability. We are updating that all the time. So there are going to be changes to this list. So if you're not sure if you're compatible with what we have, this is a good place to check and check back often because we do update and make some changes to the software and when we can get some additional compatibility. Also, we have some image application examples. So I was able to go through a couple of different formats, layouts with you today, but we do have some examples. You can download some of our layouts here. So if you're doing something similar, you can take advantage of this layout and kind of use it as a template. And we just have other analysis kind of previews to show you all the additional things that we can do and kind of the advanced capabilities of FCS Express with your data files. And then here, if you are working with the HCS Studio in FCS Express, we do have some application examples, exporting instructions if you need to know how to export those data files. So we do also offer a lot of support on our website. And then, of course, your greatest website is always going to be us. By us, I mean our technical application specialist team. We have an amazing team that is always ready to help out and make sure that you can get those questions answered. So if you do have any questions, please feel free to reach out at support@denovosoftware.com. We will either answer you by email, set up those one-on-one screen share meetings to give you a customized training or address your specific needs and information for your image cytometry data files. I appreciate you guys being here. I'm going to be online for the next few minutes to answer any other questions that come in. So if you have any questions, please get them in that question box and I'll make sure to get those answered. Thank you everybody for your time and attention today. I really appreciated you attending this webinar. If you do have any questions, please feel free to get them into that question box. I'd be happy to answer them online. And also, if you have any questions after today, after the webinar has closed, please feel free to reach out to us at support@denovosoftware.com. We'll be more than happy to answer those questions for you.